Can somebody confirm you can hear me?
Yes.
Yep.
Thank you. We have two. Oh, I know what it is. It's the my computer's working really slow here, so give me a few minutes here. All right, before we begin, are there any questions? I don't think I'm sharing my screen yet. Oops, where is that? We're only 77% done in the other Zoom file from the lecture. Let me share my screen here. All right, you should be able to see my now. 79% due now. All right. Um, Here we go. So today we're going to talk about Lab 2, aseptic transfers. Remember, you need to figure out what you want to do for your infectious disease project and then get it okayed with me. I want to make sure it's actually an infectious disease. And then I want to make it so it's a project you can actually accomplish. Like somebody's already asked, I think, for pneumonia. And I told them that that would be way too many agents can cause pneumonia. You have to pick one agent and then write pneumonia on that one agent. So I want to perhaps narrow it down. And then there's a few categories which are very popular that I tend to split up various ways. I think one person asked for malaria, and I asked them to pick a specific species that causes malaria and then write on that malarial disease. Any question about any of that? Let me remind you, you need to read through textbook on chapter 3 through the end of the section on compound light microscopy. This must be read before beginning labs 3 and lab 4, which we'll cover next week. All right, no questions. Let's go ahead and pull that down. 90%, so my computer's still going to be slow. It's the worksheet. There's module 2. Lab module 2 is on aseptic transfers. Upon successful completion of this lab, you should be able to understand the purpose of using aseptic transfer techniques, be able to determine if aseptic transfer is used from the data provided, and then lastly, be able to define the following terms. What is ubiquitous? What is an aseptic transfer? What is an inoculation? And what is a contaminant? Any question on anything so far? There are some video demonstrations in this lab. In our local environment, microorganisms are ubiquitous, meaning they're found essentially everywhere. This is problematic when working in a microbiology lab because we generally want to work with a pure culture, a culture that contains one and only one species of microorganism. Louis Pasteur developed the technique of making aseptic transfers to minimize the risk of culture contamination. He also developed a technique for obtaining a pure culture using limiting dilution. And what he would do was he would have a liquid solution containing various microorganisms, and he would then make dilutions of that solution and make multiple dilutions at different dilutions, making 10 replicas at each dilution. And if ever he got a dilution that only had one of the 10 tubes with growth in it, he correctly concluded that that one tube out of the 9 tubes that had no growth in them was likely formed from a single cell, giving rise to a pure culture. That was a very difficult way of getting a pure culture. And we'll talk about how um, Dr. Kaur, am I saying that right? Came up with an easier way of obtaining a pure culture, and he simply streaked out his bacteria on a plate. And if you streak it out on a solid plate and you streak it out 2 times on 2 solid plates, meaning you send the cells that you're trying to purify through 2 colony isolation events, there's a very high chance that you'll get a pure culture. Any question about any of that? Anyways, we're going to talk about how you can use aseptic transfer to minimize the risk of contaminating your culture. We'll learn how to make aseptic transfers in this lab. During inoculation, never lay your test tube cap or your petri dish lid on the workbench. If you do, microorganisms can be on this part of the cap or that part of the lid. And then when you put the lid back on, the microbe can fall off the lid or the cap and into the culture. So you don't want to ever lay it down. What you want to do is always hold them. Like when you're inoculating a petri dish, you should actually hold the lid over the dish. And that does 2 purposes. First of all, you're not contaminating the lid. And then second of all, you're partially protecting the petri dish underneath, and that is if something falls down from the air, it's likely going to fall on the top of the lid. It won't get in the dish, and that'll keep the dish sterile. To get the dish contaminated from the air, you'd have to have something fall only in this region here, the open portion of the dish. The rest of the dish is protected. And then with the cap, you hold the tube at an angle and hold on to the lid with one hand, the hand holding the loop, and then you put the loop in the container. The reason for doing that is you're not contaminating the lid this way. And then the other reason is when you're holding it at an angle, let me try and demonstrate a little bit. Can you see this? Anyone? Well, hopefully you can see this.
I can see your cup.
Yeah, the cup at an angle. Pretend it's a tube because I don't have a tube, and pretend this is a, a loop because I don't have a loop. When you're holding it at an angle, you have less surface area for something to fall down into the tube. It has to hit directly in this region. It have less surface area than if you hold it that way. So you want to hold it in an angle, and then there will be less surface area for the microbes to actually fall into the tube. Any question about any of that? Work efficiently. Do not talk when you're working. You don't need to wear a mask, although a mask will prevent you from breathing on anything. But the thing is, the air is contaminated already. So a mask won't help you that much because most of the things falling out of the air are already in the air. So what you want to do is work quickly, work this way so you have minimal risk, and then quickly put on the lid, and then you don't have any further risk. You also want to disinfect the workstation beforehand so that in the workstation beforehand, there's no microorganisms on the counter. And then you need to sterilize the workstation at the end as well. All right, in order to inoculate, that's transfer cells to a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. So obviously, you want to put your organisms in the sterile media here. It's been autoclaved, and so the inside of the tube is sterile, the top of the lid is sterile, meaning the inside of the top of the lid is sterile. You will need to sterilize the loop right here, and you sterilize it by either putting the wire portion of the loop inside the flame or inside the back to incinerator until the wire portion of the loop is red hot. You then cool the loop and then pick up your cells from the stock culture by taking off the lid, holding the lid in your hand, holding the tube at an angle, putting the loop, which has been sterilized, into the stock culture, picking up some cells, uh, putting the lid on the stock culture, taking the sterile media in your other hand, holding the tube at an angle, pulling off the lid, and then putting in your loop containing the cells from your stock culture. So everything should be sterile beforehand, including the wire loop, the sterile medium, and obviously the stock culture is not really sterile because it only contains one cell, the cells of your culture. All right, any question about any of that? So learn the terms of this lab. You're not going to be physically performing any of these activities because everything we do will be online, but you will be watching videos of some of these activities, and we expect that you will know and understand the material in these videos and in this lab. For procedures 1, you're going to be transferring sterile liquid into a tube using a glass pipette. And you can watch the following video demonstrating proper aseptic transfer by watching this video here. In procedure 1, you're going to transfer 1 ml of sterile water into a tube of nutrient broth. There's a problem when you're transferring water, and that is the water does not sit in the pipette at a straight way across. It forms a meniscus in either a burette or in the pipette. And that's because the water adheres to the sides of the tube. You might remember water has adherence from the lecture. And that makes the meniscus where the water will be higher up at the sides than at the bottom. And that's because water is attracted to the glass of a pipette or even the plastic of a pipette. It's attracted more to glass than it is to plastic, but you still have a meniscus on a plastic pipette as well. So where do you measure the water? Do you measure at the top? Do you take the average? Do you take it at the bottom? We always measure the meniscus from the bottom. So that would be 1 milliliter there. Any question about any of that? So you transfer 1 ml of sterile water into a tube, and there's the tube we're putting it in beforehand. And then you take that tube and you incubate it. And then you take a look at the results after, I think it's a day of incubation. And if the nutrient broth is clear after a day of incubation, you take a look at this table to get the results and then the conclusions. If the nutrient broth is clear, then no organisms are growing in the broth and aseptic transfer was successful. If, on the other hand, for procedure 1, if the nutrient broth is turbid, meaning hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. Let's scroll down here. P1 stands for procedure 1. There's the tube for procedure 1. Compare that for the tube with the broth beforehand, which is right here. And go down to the questions. Question 0, you first have to describe the initial clarity of the nutrient broth prior to inoculating, adding the sterile water or the paper disc. And was the broth clear or turbid? Write that for answer 0. And then move down to 1, which is for procedure 1. Describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees C. Answer whether it was clear or turbid. And then based on these results, what conclusion can be drawn? Your conclusion must use the wording similar to the table above. So let's go to the table. Here's the table for procedure 1. If the nutrient broth is clear, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. You can either use the exact wording here or similar wording, but you must discuss similar wording where you're talking about whether there are organisms growing in the broth and whether the aseptic transfer was unsuccessful or successful. If the nutrient broth was turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. Everyone have any questions about procedure 1 and question 1A and 1B? Let's move down to 1C now. List 3 sources of error with aseptic transfer that could explain why the nutrient broth showed growth of microbes in the tube. So you need to get 3 reasons, and they have to work for procedure 1. Like sometimes I get students saying something like, well, the needle that you use, which we didn't use because we didn't use a needle, we didn't use a loop either, wasn't successfully sterilized, and so that contaminated the tube. You didn't use a needle, so that's not a correct answer. Any question about any of that? So give me 3 reasons how you could have got contamination. All right, then let's go to procedure 2. In procedure 2, we're doing something similar, but instead of transferring 1 ml of water, we're going to transfer a sterile paper disc using clean forceps, using proper aseptic transfer. Also watch the video for procedure 2, but you don't need to watch it a second time. Procedure 2, you're transferring a sterile paper disc into a nutrient broth. If you were to come into a lab, we have a petri dish containing this sterile paper disc. There's the table you have to use for procedure 1 and 2. And there's the picture for procedure 2, the tube. You can actually see the paper disc on the bottom of that tube, right there. And then answer the questions for procedure 2. For result 2, describe the clarity of the nutrient broth after inoculating with the sterile paper disc and overnight incubation. Was it clear or turbid? And then for 2B, based on the results, what conclusion can be drawn? Your conclusions must use wording similar to the table above. So answer that there, and then you're done with 2. So let's go to procedure 3. In procedure 3, you're going to be transferring Serratia marcescens to a broth. Remember, a broth is a liquid culture. We have a video for you to watch for procedure 3 and 4 to how to correctly transfer from a bacterial stock culture to sterile broth and to a sterile agar slan, which will be what you're doing in procedure 4. So watch the video. You only need to watch the video for 4 minutes and 23 seconds into the video. In this video, bacterium E. coli is transferred in the video. In our procedures, 3 and 4, we're going to be transferring Serratia marcescens. Serratia marcescens is a pigmented bacteria. E. coli is non-pigmented. That's a difference in the look, and so you can answer some of the questions from the pigmentation of Serratia marcescens. There's also an error in the video. You should look for the error when the instructor puts E. coli into the loop into the stock culture of E. coli, you can hear some sizzling between 3 minutes and 5 seconds and 3 minutes and 20 seconds. Do you know what that error is? So you should actually look for that error. I'm not going to grade you on that, but that's an error you should look for. In procedure 3, you're transferring Serratia marcescens into a sterile broth. Broth cultures are problematic because it's difficult to determine if contamination has occurred because the cells do not form colonies when they grow up. They will grow all together and mix up in the liquid culture, and so it will be difficult to determine if there is contamination in a broth or a liquid culture. You're not going to be able to detect the contamination unless it is a major contamination. If it's a minor contamination where the contaminant is not the main cells in the broth, then you will not be able to see the minor contamination because the color of the broth will be dependent on the majority of the cells, which in this case will be the cells that you inoculated into the tube, the ones you want to grow. The contaminant you will not be able to see because the contaminants will be hidden in the broth from the main cells, the majority of the cells. Any question about any of that? Once again, you use this table for getting your results and conclusions. And there's the tube for P3 right there. Going down to question 3. Results of P3. Describe the clarity of the nutrient broth after inoculating with Serratia marcescens in overnight incubation at 30 degrees C. Was the tube clear or turbid? 3B. Based on the results, what conclusion can be drawn? Your conclusion must use wording similar to the table above. So for procedure 3, if the nutrient broth is clear, then no organisms are growing in the broth and aseptic transfer was unsuccessful. Why, if the nutrient broth is clear for procedure 3, are we saying aseptic transfer was unsuccessful? With procedures 1 and 2, if the nutrient broth is clear, we were saying aseptic transfer was successful. Why the difference? Anyone? Anyone have a guess?
Because you're trying to isolate that one specific bacteria
Yeah,
for
in
procedure
procedure
3.
3, you're putting one specific bacteria to the tube. So we expect that one specific bacteria to grow, and then the broth should be turbid, hazy or cloudy, and organisms should be growing in the broth because you inoculated the tube. In procedures 1 and 2, you put sterile water in tube 1, and then in tube 2, you put a sterile paper disc in. So we expect no organisms to grow in the broth because if everything was done aseptically, the organisms should be growing in the broth for procedure 1 and 2. But in procedure 3, we're actually inoculating cells in. So we expect that the organisms will grow in the tube. All right, for 3C, how do you know whether you've transferred the correct microbes, Serratia marcescens, into the tube? And there's actually an easy way to determine that, and it's because Serratia marcescens is pigmented. All right, let's go to procedure 4. In procedure 4, you're going to transfer Serratia marcescens onto an auger slant. Serratia marcescens, when it grows on an auger slant, will be colored red, pigmented red. If it grows in a liquid culture, it'll be pigmented pink, by the way. So it's not quite as intense when it's in a liquid culture as when it's growing on a slant, solid culture. Contamination on a slant can be more easily detected in a broth. What you want to do is inoculate the slant, shown here, in an S-like movement. So you're inoculating this region of the slant there. And if you have growth only on the slant and it is only pigmented pink on this region of the slant, then you know it is what you inoculated. If on the other hand it's growing off the slant, so let's say it's growing right here on the side where you did not streak, or it's growing someplace where you did streak and it's the wrong color, it's not pigmented, or pigmented another color other than red, then you know it's a contaminant. And it'll be easier to determine because it will grow as a colony. So you'll see either we'll say a non-red colony growing right there, or you'll see a colony growing off where you streaked it, and that will be a contaminant. So it's easier to see a contaminant when you're growing on an auger slant than when you're growing in a broth. The only way you can determine if you have a contaminant in the broth is if the contaminant makes the broth the wrong color, and you usually won't be able to determine that unless the contaminant is close to, if not the major cell, in the tube. All right, so here's the results for procedure 4 here and the conclusions. Here is the result for procedure 4 there. You'll note that there's, sorry, so much growth here that the entire region of this region is filled in totally. So they did streak it this way, and there's so much growth that just grew in this region confluent. But if you look real closely, let me blow this up way big. Scroll down now. You can see a little bit of auger right there and a little bit of auger right there. There's no growth in this region, so there's no contaminant. There is growth in this region here, and then something looks funny there. What happened here is that there was water in the bottom of this slant, and that's why the cells from here went over to here and there. And right there, you see this lighter color there? That's actually a drop of water right there, why this region looks different. So that's not a contaminant. That's a sterile drop of water, and the water is spreading the bacteria all over in this region. And earlier, the water was all over this region, and that's why there's cells still here and here. So that's not a contaminant either. And as you see, there's red everywhere. That's not a white colony. That's actually the light being reflected back. So there's no non-red colonies here. Of course, it's growing confluent. You can't see any colonies. But the point is you can determine even if there's minor contaminant on a slant. And in a broth, you wouldn't be able to determine that. Here you can see the broth in a liquid culture looks pinkish in color. On the slant, it looks reddish in color. Shrink that down a little. So answer the questions to 4. P4, is there bacterial growth on the slant? Just answer yes or no. How do you know? State why you know. And 4, does it appear to be the bacterium you're inoculated or a contaminant? And then why? I'm wondering if I gave you another question that I don't have here. Let me take a look at procedure 3. Nope, I don't see it there. Okay. All right. Any questions? If not, go ahead and get started on the lab. I will be here until 8 o'clock to answer any questions or until the last student logs out. So if you have a question, make sure you ask before the last student logs out or before 8 o'clock. After 8 o'clock or the last student logs out, I'm going to log out as well. But if you have any questions, I'm here to help you.